Testing, testing. Testing, testing. Testing, testing. One, two, three, testing, 1, 2. Audio seems to be okay. Okay. Hey, Paul. How's it going? Finally got some water samples today after a special trip and my new gadget to collect water from a distance. And this is what we currently have. I'm actually not 100% what it is, but it caught my eye and I'm checking it out. And without the border around it, it looks like a ball of individual phytoplankton, like maybe like a volvox, but it's too small and too defined with that border, so I'm going to zoom in here. See what we can see. Might actually be a diatom, of one of those weird radial diatoms. Oops. Let's get the light fixed on there. And I'm going to zoom out on this guy because that's too close. Put a little too much water in the slide, so every time I bump the table, it sloshes around a little bit, unfortunately. But yeah, it's a pretty good looking little specimen there. At this level, it looks pretty much like a radial diatom. But again, I'm not totally sure. I'm gonna have to look this one up. Let's change the color temperature a little bit there. Hello, Teeny. How you doing? Just got some new water samples here at the local salt marsh. Just investigating some things I haven't actually seen before. So a couple of these I'm gonna have to look up later. I'm sure, this one for sure. First I thought it was colonial phytoplankton, but the structure here looks really, really a lot like a radial diatom of some kind, but then it doesn't have the lines I'm used to seeing on a radial diatom. So, really interesting structure. Let me fix that contrast. It's a little too... It's not as sharp as I would like it. Come on, let's go. There we go. A little more contrast. Yeah, it's really interesting structure there. A lot to focus through. It's very, I thought it might be like a disc shape, but it appears to be more spherical because there's a lot of stuff to focus through. Yeah, I remember you from Microbe Hunter. It's got a great channel. I can only aspire to be a fraction of as good as Microbe Hunter is. But we have a little bit different focus. His does a lot of stuff on slide preparations and growing organisms and doing some other stuff. I kind of randomly grabbing stuff from the wild from all over. Plus I also do stuff with plants, which he occasionally does as well, and do stuff on fungal spores and bacteria and weird kitchen chemistry stuff and all that. Kitchen microscopy. Anyway, so this is a really interesting little critter here, but I'm not sure what it is. So I'm gonna have that cataloged on the stream and then go back and check it out later. Let's see what else is here before the slide starts to dry up. Alright, let's fix this auto thingamajig, ain't working too well. I'll turn that off. Okay, let's recenter everything. This is off kilter now. And let's see what else we got here. Survey the slide. So this is at the end of the stream. I haven't had a chance to organize the pictures yet. I literally came right in from the field with my samples and started doing this. So I took some pictures at each station. I took these samples just to give an idea of the habitat. And at the very end of the stream, before I turn it off, I'm going to quickly post the pictures in the stream so you guys can refer to it. I'll put chapters in later, as I usually do, so you can jump to the area where I collected them, as well as the general view of the salt marsh preserve where I got these from, and just give you an idea where they came from, a sense of place. All right, so there's nothing there. Let's go back to got more tissue bits here, plant bits. There's another one of those little structures there. First I thought that might be an amoeba, but it's not moving. But it sure looks like an amoeba spread out. Let's take a close look. Nah, it's something else. This auto adjust is not working tonight. It's not being my best buddy here. Come on. There we go. That's a little bit better. I can fix it with this. Yeah. There we go. Fix that contrast. Yeah, it's an interesting little. It's a very thick structure, very polyp-like, but it's definitely not an amoeba. I'm not sure what this one is either. That's some weird samples here, for sure. Interesting. Okay, so I'm going to move on. That's just an interesting cluster of stuff. The damn auto thing is not behaving. Come on. These days I'll tailor my OBS to make it a little more transparent. You don't have to see all the sliders I'm adjusting. Okay, let's do a little bit of a zoom in here, just because we can. Looks So let's continue to explore more marginal stuff here, just materials, plant bits, nothing swimming, nothing darting about. So far the sample's pretty dull. I did stir up the area a bit as I collected, so I'm getting a little bit of benthic stuff as well as stuff at the surface. So anything is possible. There's another one of those little cluster things. Whatever the hell that is. Let's fix this again. This is too bright. Come on. Okay. So that one's not very interesting. There's a piece of a plant there. What else we got? Okay, so this slide is pretty much a wash. Should usually find some within a few seconds, because where I collected from is very rich in all kinds of goodies. So that was sample D. Let's try sample C and work my way back here. See what we can find. I'll catch up on the chat in a bit here. I gotta get some of this stuff prepped. Come on. Sometimes these cover slips, they come brand new, but they're cloudy. It's really obnoxious. So I have to actually clean them before I can use them. Alright. This one has a little bit of color to it, so it might have some fragments of all kinds of things. Oh yeah, there's all kinds of debris in here. Should be promising. I'm already seeing, looks like some diatom tests, little shells in there. So let's zoom out here a tad. Let's fix that thing. I had the wrong dude in there. Let's put the other one in here. Let's see if I can get that to behave correctly. No, that's not behaving correctly. Let's try this one maybe. I might have the condenser messed up as well. There we go. Condenser was not set right. So now we can do some dark field as well as some oblique lighting. Let's try, what do we see here? No crazy move. Some little flagellates here bouncing around. Let's jump in a bit. Fix this. Fix this. Okay, where do they go? Let me zoom out here a bit. Hey Gary, how's it going? Sorry, I'm behind on the chat here, just trying to find some specimens here and some new water samples. Come on. Saw you guys bopping about earlier. Where did you go? Oh, they're in a different focal field. Okay. I think I was down at the top of the cover slip. Alright, there's some diatom tests in there. Little cigar-shaped ones. And you can see all kinds of things flittering about in there. But they're very, very tiny. Let me go in another level. Let's turn the oblique lighting off. And go in for the traditional bright field for now. I know Sasquatch has been in the Kent's pub for a while, Paul. In fact, it got a little interesting with the last ones he showed up on. So it actually ended up getting deleted, I believe, because of some drama. But you didn't hear that from me. There's an interesting little structure here. But I'm looking for live things. Where is some live stuff? This is just tissue. Lots of tissue, lots of fragments. Making a lot of life here. Let me go back a level. It's too close right now. There we go. Hey, got a ciliate. Holy smokes. Actually, no, that's a flagellate. That's a euglena. Go, euglena. Yay. Alright, now I gotta track him down. This is the video game aspect of it. So you see he's got a bit of a greenish tint. He's got a red eye spot. Let me just zoom in a bit on here and fix this. This image is too bright. Look at that guy go. I don't want to get too, see how he's really fast sometimes. I don't want to get too damn close and then it's hard to follow. And I didn't put any methylcellulose on here to slow him down. So, let's see what it does for a bit here. You can watch the behavior. It's quite active. Whoa, 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 where'd you go? There you are near debris here. That's a really nice euglena close-ups in some previous live streams if you want to see them up close and personal. You can see the contents inside the cells shifting around and all that. And they can really, they're super flexible. They can distort like crazy, but kind of bunch up into almost like a ball shape. And then go the standard tapered look. There's one of those huge diatom shows right in the middle right there. Those are beautiful when they're alive, swimming around. Oh, there's an oscillatoria. Blue-green algae there, and it's moving. Because they can move. Nice greenish rod. Let me give you some more light there so you can see the color better. Let's see if I can go to dark field again on this guy. Get some color. So that's oscillatoria right there. I'm getting too much stuff here. Let's see if we can bring it down a tad. Some natural light with the dark field. So really nice oscillatoria bunched up with some diatom shells and some debris there. Let's go try to find euglena again. Where did it go? Whoa, nematode. There's a nematode having a fit. This place never lets me down. I always get good specimens out here. It's a very rich, fertile salt marsh out there. Alright, let's change this a tad. Can make it a little bit different. That's about it. All it's going to do for that. So yeah, it's in a little debris there. But it's really nice on the dark field. You can see the nice transparent body and then all the contents in the middle. And all their transparencies as well. In a bright field, it looks mostly like dark material, not that much detail usually, but they really shine on the dark field. Oh, what do we got here? Is that a Euplodes? I think that might be Euplodes. They move around like insects, even though they're single-cell protists. See how they crawl like that? They have these thickened bundles of cilia called cirri, and they crawl on stuff like little beetles. It's the craziest shit. I love these guys. And you can see the size in relation to the nematode. They're not tiny, but they're fairly small. And it's moving like crazy. So I can try to get closer to it. Oh, there's another one next to it that's slower. This guy's a little easier to track, I think. Let's zoom in real quick and see if I can still follow it. There it is. Now you can see it crawling on stuff. Let me fix the brightness and such. It's not behaving. There you go. You can see that walking on the cirri like their little legs. This objective needs to be cleaned or something. It doesn't look so good. But I love these guys. They're so much fun to watch. Because they act like something that's multicellular. But they're just single cell critters. So cool. Go. Yeah, this image quality is not that great on this objective. Plus, this is a lot of debris. I have to focus in and out. It's going up and down as well as side to side. There's a nice diatom next to it. Look at that beautiful color and the oil droplets inside. Little spherical oil droplets. A little more light. A little bit more light. There you go. Get some color in the droplets. And it's also moving because they locomote as well. Yeah, this is a good slide so far. Lots of good crap on here. Yeah, diatoms are cool. I've only found a couple of the really special ones in that area a few times. I have an older video I shot with an iPhone because I didn't have a proper camera connection that I still need to upload. It's beautiful. It's just one that, it's like one of those fold-out yardsticks that you used to put in your pocket, made out of wood. But imagine the instead of being joined at like little hinge joints, they can actually slide along the long edge of the joint of the structure. It's this really cool colonial diatom. They're really interesting. Oh, there's Euplodes next to it. Yay, go. Actually, you know what? This one doesn't have the curvature of Euplodes. This might actually be a Stylonchia. It's got the longer cirri at the poles, up the north and south. Head and tail, for lack of a better description. But same idea with the cirri. Really long, bristly cirri, top and bottom. And then some kind of regular cilia on the sides. But yeah, these guys are also fun to watch. Very active. Euplodes usually has concave side. kind of a curved back. Again, for lack of a better term, they don't really have a back, but it looks like a beetle on edge. And the Siri come down and they just walk over things in all different directions. Like they're walking on a log or a small rock or something. Very cool. And you gotta focus through all this debris because they're going crazy here. Got a lot of detail inside the cell there, but they move so fast you can't really see it too much. They just take off. So one day I'm going to have to put some methylcellulose on here and slow them down so they have a bunch of debris to swim through. But yeah, see, it uses the Siri like little legs, pushing along material. So cool. Have you seen these, Gary? These are really interesting little critters. I like these guys a lot. I see them a lot at this site where I collect. Now it's up and down a lot. I gotta focus in my Z too much. Let's go somewhere else. What else we got? There's a large dead diatom, the big boxy type in the middle. And you can see the, as I focus through, you see the, looks like a comb, kind of filter comb thing on the edge there. That's the, what they call the Rafe, or Rafey. And I believe it has to do with their locomotion. They're still kind of understood about how they locomote. They used to think it was some kind of ion pumping, and then now they think it's some kind of a mucilage they exude from there and push along. But I read some other stuff lately that said it was something else. I don't know. I haven't kept up with that. But they're really interesting. As you focus through, you see those structures in the glass shell left behind. There's this dude again. There's another diatom. This one's more of a fat cigar shape. Get quite a variety of diatoms out here at this site. You get the long tapered cigars. You get the cigars with the bent ends in opposite directions. Ooh, was that a... Look at that. There's a diatom right there. This one's got, I call the smile. It's kind of bent, or like a smile of the banana. It's kind of bent in one curve and concave on the other side. It's moving through debris though now. Come on, banana, get out of there. Need some more light. And some more contrast maybe. Can I do some dark field here? Or oblique lighting? Let's do a little oblique lighting in this guy. Yeah, it's not working too well. He's in all that debris. It looks like a mess. Let me switch back here. Sometimes bright field is the best. Oh, and there's some filamentous colonial cyanobacteria there. See, those are the little clear, thin rods there. They're quite long, moving junk around. Those are basically prokaryotes, like bacteria. Is that guy coming out yet? There, he's coming out a little bit. You see kind of a banana shape there? It's curved on one side, more flat on the other. It likes that debris for some reason. There's that filament moving through. So that's the banana. Let's see, keep going here because I'm sure the slide's going to start to dry up as I usually do. There's another filament cyanobacteria thingy there, bouncing around in that debris. What else we got? More diatom shells all over. There's one of those big diatom shells poking through. That's the tapered one I'm talking about. See how it's tapered on one end, curved one way, and tapered on the other end, curved the other way. Those are cool too. I like those diatoms a lot. That's just the shell left behind in the middle there. So curves one way to the left and curves the other way to the right. I have to kind of focus through it to show it to you because it's at a different angle. And here's another one, which you can really see the rafe there, the teeth. It's like a tooth look on the edge of the shell. And there's a banana one right next to it at the top of it. It's dead. And what else can we find here? Lots of dead diatoms. There's another Silatoria moving slowly, like the Imperial cruiser from Star Wars, or Imperial Destroyer, or whatever the hell it's called. They're just very fun to watch, very peaceful. You can see the banding on it. The stripes going perpendicular to the length of the body. There's a nice shot of the texture there. Can't quite make out the cilia, making them beat. Or in this case, I think they have tiny flagella, not cilia, but they're very, very tiny. Can't really see them. But occasionally get up close and you can see some stuff going on inside. There's a little bit of movement if you look carefully. And a lot of the microscopic plants, the single cell plants and stuff, you get cytoplasmic streaming where the chloroplasts move around in kind of a circular pattern inside the cell, given that there's room to do so depending on the shape of the organism. But there's definitely some kind of movement in there if you look carefully. There's some action happening. I don't want to go too much closer, then I have to put the oil lens on, and it's a big mess to clean up, and I'm still changing powers a lot, so I don't want to do that right now. Might be able to, if I can make this a little brighter. And yeah, it's as good as it's going to get focus-wise. Obviously it looks better in the eyepiece than it does on the camera, but looks pretty decent. This is 400 power right now. Sorry, I'm missing some of the chat here. I'm just engrossed in trying to find some goodies here. I forget to check sometimes. Let's go back to dark field. There's a Silatoria again, looking nice. Dark background. Let's see what else we got here. There's some tiny little flagellates going crazy in the background, but they're just too hard to follow. There's another long tapered diatom. Oh, wait, what do we got here? What's happening here? Is that another nematode? Yeah. Nematode on the debris here. Let's go. What is that? Big chunk of debris. Yeah, all kinds of diatoms. Diatom shells here and there. More Silatoria. Oh, what was that? Debris. So I'm gonna find a rotifer or something, but so far no luck. But I got plenty more samples to look at. I sampled from 4 different sites at the one site and drove down the road. This is a very stubby oscillatoria. Very stubby. Let's fix that lighting a bit. It's kind of cool in 3D, but if you want to see more of it, you gotta give it a little different treatment here. Let's see it pop off the background there. See the texture of it. Yeah, Raspberry Pi Cam, I haven't played with those much. I play with the Pis a lot for other projects, but this is just one of these, it's similar to, it's like the Swift / Amscope 15 megapixel cameras. It's halfway decent. I'm saving up to get a really nice, I was going to get a DSLR, now I'm going to get a mirrorless because my other channel I'm starting up is an astronomy channel and I need a really, really fast camera. I need 30,000 ISO or higher, high quality, and I'm buying a Meade Astrograph f4, f8 Newtonian telescope. Those two together will allow me to do literally galaxy shots in minutes that I can actually share online, do live stream as I'm taking the pictures. So can't wait to do that. It's pretty special because up till recently, unless you had all the right stuff, you had to basically do long exposures or stacking exposures, and it could take you like half hour of imagery or 200 5-minute exposures stacked together and all that kind of stuff. So I just don't have the time for that, and also can't do anything live with that. So I'm all about the live streams when I can do it. So I want to do an astronomy live stream because no one really does those. Everyone does mostly prepared videos with long exposure pictures and repurposing other people's content. I want to make my own content. So there's another basically dead large diatom there in the middle. Those long cigar guys. Another oscillatoria, another oscillatoria. Oh, there's one of those little dudes again. That might be the Euplodes. This is more round. Let's see. Let's see. Let's get that lighting fixed here. Anyway, that camera will not only be a kick-ass for this, but really good for astronomy. So that'll solve all my problems. But they ain't cheap. Well, it seemed to have taken off. Or is it a different focus? A different focus, of course. There you go. Okay. Yeah, you see how this one is a slightly different, this is more disc-shaped. And as it rotates, it's more concave. This is Euplodes right here. This guy's cool. Also crawls around like a critter, like a little insect on things, even though it's a single-celled critter. And it's mostly spiky at the back. As you can see, the cirri are mostly in the back. And on the bottom, it uses his feet, like little legs, to crawl on things. And these guys are so much fun. And as they rotate, you actually see the concavity. They actually have a kind of a curved back shape and then a concave other side. And when they, which is perfect, allows them to kind of walk on a surface just like he's doing right there. Boom. Just like a beetle would do on some leaves or branches. So much fun to watch these guys. This is back to a 400 power again, so they're pretty small, but totally worth watching. And they tend to crawl around a small area like this, so they're not too hard to follow, even though they move pretty quickly. They don't just like dart around swimming. Their behavior is more to crawl like this. Crawl and walk on things, which is so weird for ciliates in general. So they generally just swim all over the place. this guy loves to crawl and walk. But it's in and out of focus. Oh, there's two of them now. It's in and out of focus a lot because it's constantly going up and down the Z. It's very busy. But they just, their behavior is fun to watch. They have this kind of cute little appearance. This little kind of disc-shaped thing with his little bristles coming out the back. Now they're tag teaming on the oscillatoria. Now he's walking on. Look at that. That's what I'm talking about. Walking on the oscillatoria. So cool. And now in 3D, he's walking on the back side and on the front side, so you can see it through the oscillatoria. Love that. Yeah. This is good stuff. Just like a little beetle. Or ladybug or something, you know? So cute. I've never seen any other cilia do this. And in all my years of looking at stuff in scopes here and there, it's the only one I've seen do this, period. So cool. It's gonna follow it up and down, up and down. And the nice thing is it's confined to this rod for now, so all I just do is move a little bit to follow it. And it's a predictable path, so I'm not losing focus too much. But hopefully I don't get seasick watching this because I do have to move back and forth a bunch. It does occasionally stop, you get a nice little shot of it. And then it just moves around, but I love when it goes behind and in front and you can just follow it. The transparent algae there. And of course it's being magnified by the shape of the algae. So cool. It's algae lensing. You can see the other one in the background moving back and forth, but it's quite a bit further back in the Z, so I'm not going to try to follow both of them. It's just too much. Back down again, back up again. Gotta wonder what it's doing because I don't imagine it's getting anything off the oscillatoria. It's just using it to explore. Oh, now they're both on the same thing. Yay, I was waiting for that. This is like when you get little birds like woodpeckers and brown creepers and stuff on the same tree. So cool. Up and down, up and down. This is great. This is some of the best footage I ever got of these guys. So much fun. How can you not love this little guy after this little show it puts on for you? Oops. Oh, Gary, so I have my original channel, before I started this one, as a music channel. All my channels have addict in them. So Synth Addict, one word, it's music, production. I even use some of this footage in a couple of videos for some music contests that we have, this weekly jam, hardware jam thing. So I pulled some clips out of my live streams and made kind of a synced piece of music. And even one of my really weird pieces of footage I have where one of the ciliates, I think it's a Styloncia, basically disintegrates on camera. I don't know if you ever found these guys, when sometimes they're just swimming, That Microcosmos channel has some amazing footage because that guy's like the best ever. And he had a really cool thing about death of a ciliate, and I was like, wow, I've never seen that before. Sure enough, a couple months later, I have footage of the same thing, just not nearly as good as that guy's stuff. That guy James is like the master. Some of the best microscopy I've ever seen in my life. But anyway, he had this footage, and so I had this creepy music I made for this thing, and then I ended the piece with some effects as the thing disintegrated, so the audio kind of went with the death of the poor ciliate. That's on my channel. And then I started an MicroAddict channel. Unfortunately, there's already a guy named MicroAddict, but since I have a theme, I'm not changing it. So he has a big established channel with lots of produced videos and stuff, but as far as I know, he doesn't do any actual live streams with his telescope. He just has produced videos. So I'll be the MicroAddict that does live streams once I get all my crap together. Those are my 3 channels for now. Right now, the MicroAddict has no content on it, really. Oh, look at him hitting and spinning. Go, go, go. Sometimes you'll get them doing that. They'll just sit in place and spin. Oh, now you're back. Go, go, go. Yeah. Some of these fine controls are sometimes a little too tight. Things jump around. Yeah, I love this. Guy going up and down this microscopic tree. I think I have the links in... I definitely have the links for this channel and my synthetic channel. I don't know if I have the other ones in my... It's a good point. I'll have to check my banner and see if I got them all linked back and forth. I actually hadn't have any time to do that. I did one of these last night, kind of impromptu, without water samples because I couldn't find any close to work yesterday. But then today I thought I'm going to go out of my way, get some nice samples, and do another show 2 days in a row. This guy's a trip. Too funny. I could stay him all day, but there's more stuff to do. Plus the slide's probably going to dry out soon. I better mosey on to another area of the slide. There's a shot for scale. You can see how small they are in the lower power. This is 40 power. No, sorry, 100 power. 100 power. What else we got here before this slide gets old? There's another one of those little egg-like things, like a Death Star. I'm not sure what that is exactly. Always something new to discover. It's like the Saturn's moon Mimas. It's a natural version of the Death Star. Big ass crater on it. And a little dude right next to it. What is that guy? Little crazy flagellate popping around. Ooh, it's actually changing shape. That might have been a contractile vacuole. That thing in the center closed. Oh, no, maybe it just shifted a little bit. Definitely something interesting. And the flagellate's back. Alright, let's go looking elsewhere here. Oh wait, that thing is actually moving a little bit. It's weird. It could actually be one of those large vorticellas collapsed on itself. Sometimes they'll ball up like that and then open up again when they detach from their stems. Not quite sure what that guy is, but there's some activity inside for sure. I'm seeing some movement dancing around in there. It's not just Brownian motion. That little flagellant's fun. He's kind of spinning around. Go, dude, go. Alright, well, time to move on to more goodies. Got more samples to look at, so I better hurry up through this. What else we got here? Oscillatoria. Diatoms everywhere. Oscillatoria everywhere. God, there's tons of them in here. More debris. More diatom shells. More tissue. Yeah, you can see the water starting to dry up a little bit, getting some crystallization there. Oh. Ah, it's that little... That looks like the, I think that's the Styloncia dude we saw before. Going to town. Different shape than Euplotes, but... Alright. There's that long tapered, curved end diatom in the middle again. Let me fix the lighting here. Yeah, it's not a very good view of it anyway. Alright, let's try another slide. That was very nice. That was collection site C. Let's go to collection site B. Okay, that was C. Now we'll do B. Get my cover here. Yeah, it could be a cyst of some kind. Alright, let's go. Site B. What the hell we got here? Oh, nice. Nice. Let me fix that. Can I get a dark field? Yes, I can. There we go. Let's zoom in on that dude. And fix the focus. And very nice. I haven't seen one of these in a long time. This might be an amphileptus, I think. I'd have to look it up again. It's got that funny tapered shape. Let me fix the light. It's still a little bit too dark. Oh, there's some oblique lighting on it. They get a little 3D effect. Nice 3D. There we go. So it's got a little snout thingy that's seems to be a bit flexible. Not like there's some other ones out there that use a literally flexible snout for snagging prey. I think this is Amphileptus. It's been a long time since I looked one of these up. But very distinct shape. Kind of teardrop shape. It's also going in and out of Z a lot, so I'm having to constantly focus here. These guys love debris. So just like if you're into birding or you're into certain types of mammals and stuff, you go out in nature walks and stuff. You tend to find they like to cluster in the little protected areas like a forest or a bushy grassland or something like that, and that's where you find a lot of the activity. Same with these guys. In this case, they're not hiding so much as that's where the goodies are, apparently. Tunneling through all this debris looking for yum-yums. But the idea is similar. You're just looking through patches of debris. I never totally bypass a patch of debris. I'll stay on it for a while, waiting for stuff to pop out or go in and out of it, because look at this guy. He loves it. Going crazy for wherever this debris is. Oh, that was cool. It just went around it. You can see how flat it is. It just did a little, look at that. Now it's following the profile of the debris like a snail. God, that is so cool. Almost like a planaria. Look at that. That is really cool. So like the Euplotes was crawling, this guy is slithering over the debris. Like a slug. That is wild. Never seen this behavior before. Fix the brightness a little bit. It's a little too bright. That's a little better there. Yeah, this is really cool. A little more contrast, a little less brightness. That looks better. So yeah, it pays to hang out in these little areas sometimes. This guy's going nuts for this debris. He's crawling all over it. Look at that. You can see how flat it is. Flat and floppy. It's just kind of crawling over and kind of wiggling. That is so cool. Again, he's going in and out a lot, so I have to constantly focus. But look at that. This is the same thing I do with birds and mammals and everything else that I go out and I like to not just see them and go, yep, I've seen it. You got to sit and study them for a while because you never know what they're going to do. They do really weird behavior sometimes. And you might see something special that only a handful of people have ever seen. So it definitely pays to be patient. Although unfortunately a slide, you can't be too patient because it does dry up eventually. Look at that guy. Let me fix this a little bit darker. I have some footage of some paramecia, some of the first footage I shot since I did this channel. Kind of in a similar situation where they're kind of not bending over stuff, but kind of squeezing into crevices and stuff, and just deforming their bodies. They're actually they're rigid, but they're quite not as squishy as Euglena seem to be. But they were kind of smushing themselves into these little crevices. It was really fascinating. Seeing them kind of wedge themselves into these little areas. But this guy's just going nuts. Really flexible. Cheat a little bit and zoom with my digital zoom a little bit because I don't want to lose the field. Change the ambient preset a little bit, make it a little more interesting. Oh, something went swimming by. Oh, nice. That is something else. What is that? It doesn't have the Siri, so it's not a... Interesting. I'm going to follow that guy next. Alright. This guy's done quite a bit of fun stuff, but he hasn't done anything for a while, so look at this dude. He's on an orbit. Just follow the orbit. I love when they do stuff like this because you don't have to move much to keep an eye on them, and they tend to stay in a similar plane of focus, so they're much easier to observe. I've seen these before, but I didn't identify this one. But I have footage of two of these dying fairly dramatically on camera, where they basically stop moving, contractile vacuole stop moving, and then they sit there for a while and they start taking on water because the contractile vacuole stopped working. They start to bulge and then they literally pop, and all the cytoplasm and organelles spill out into the matrix, into the water, and it's fascinating because the organelles continue to move and kind of cycle around, and they don't pop immediately, but eventually they all pop. So it's, very, very dramatic footage, and I recognize these guys because this is the one I captured a couple times. Just didn't quite identify it yet. But I often find them doing these circular patterns. They'll get stuck on some kind of a pattern and just do this. And I have one where one of these guys was getting caught on an air bubble, and due to surface tension was kind of locked onto the air bubble and cruising around it like it was like a Ferris wheel or something like that at high speed. It was really funny. And it was going one way, then it was going the other way. I think it was having trouble disengaging from the air bubble, the interface, surface tension issue or something, cohesion. So then it finally popped off, but then another one got on there and started spinning around. It was just really funny. But these guys are interesting. They have a lot of structure inside. It helps to have this oblique lighting too. It really makes it pop. I'm going to try change the lighting a little bit. Let's see if we can do a little bit of a dark field instead, so you can get more of a transparent, glowy look to it. Now I can think I can zoom out digitally and get more image. There we go. That's what we want. I can't quite get the whole circuit, but I can get a good portion of it. Oh, now it's moving over here. Oh, this one's doing a big circle now. They're just unpredictable. You can't quite know what the hell path they're going on and when they're going to pop back in. Just doing a big old set of circles. Where the hell did it go now? Hello. Where'd you go? I don't know where it went. Just took off. So sometimes they'll do circles for a while and then just take off. Like a cat leaving a room. Just boom, they're gone. Let's look for some other thingies. Small diatoms. Planet debris. Tiny little flagellates spinning around like crazy. What do we got here? Oh, eight. Nematode. Now I can zoom back in a bit. Get a nice, better view of this dude. Again, it loves the debris. Oh, now it's gonna. Oh, not quite ready to commit. It's gonna come back. Yeah, they just latch onto this debris and just go crazy sometimes. Oh, now it's swirling around. That is cool. It's enveloping the debris like a snake. Again, it's a crazy ass behavior this night. This is nuts. I haven't seen any of this behavior from any of these guys ever. Except for that guy going in circles. Look at that. It's like a snake going around a tree branch or something. Oh, it's ready to leave. Oh, no, it's going back. Not quite ready to commit. It's got the safety of this piece of debris. Oh, there's one of those little ciliates again. Which one is that one? I like this guy doing the snake behavior. This is crazy. And now it's free floating. Ooh, there's a very large colonial algae there. Very active too. Look at that. It's moving quite a bit and also contorting, making little loops. That's cool. Bending around. Wild stuff. I'm glad I did this tonight. Got some crazy ass stuff on here. Oh, yeah, Gary took off. Sorry, Gary, I missed your comments. Yeah, Teeny, I think you might be the only one. Everyone else had to bow out because it's either late for them or they got something else going on. Thanks for being a diehard. Let's see what else is sliding. Oh, now it's doing little twisties. Look at that. That is cool. It's braiding itself. I haven't seen that behavior before either. That's wild. Let's see what that looks like in oblique. Get a little more 3D structure to it. Nice. Oh, only 1 p.m. That's cool. Yeah, it's almost 11:30 p.m. for me. No problem, because I'm a night person anyway, so it's early for me. This, whoa, look at that. Whoa, that's wild. Whoa, whoa, whoa. Very dramatic. So even this colonial algae can be interesting. Not always so boring. Alright, this one's a little more weird. Let's go back to a more typical, kind of a creepy sci-fi motif. There you go. And I got 3D with some real contrast. You can see the convulsions and how it's also sliding along itself. That is so cool. It's twisted and sliding. So damn flexible. That's crazy. Oh wow, 12 hours later for micropunter? Huh. Interesting. That is a major time conflict. Oh, he's gonna slide that thing back through and go straight. The curve is now gone. Here it goes. It's so funny, they got all this debris stuck on them. It's sticking through debris and it's got debris on the end. Big messy. Oh, there he goes. It just popped straight. Bing! Yeah, this is a colonial algae, kind of like a Silatoria, but this one's more like you can't really see the color as well. It's not as obvious, but it's a what they call cyanobacteria. So it's blue or blue-green algae. It's more prokaryote than eukaryote. So it's closer related to bacteria than to other plants. True algae. Yeah, it's going the other direction. So they photosynthesize, but their pigments are actually different, which is why it's a blue-green color. I forgot the chemistry. I used to know all this stuff a long time ago, but if you don't use it, you lose it. And really interesting critters. This thing, yeah, see, it's at a different. I had to focus a lot to catch the whole thing. It's bent in the Z direction as well, so it's pretty wild. there's, you know, not too far from San Jose, California. Silicon Valley. Where the rent is way too high, and the property ownership and everything else. Now it's really bent going this way. But the nice thing about this area is, excuse me, there's all kinds of salt marshes around here, fresh water from the features, grassland, forests, desert-like areas, scrub areas, coastal areas, because we're not too far from the coast here with the beaches and stuff. So you have kelp and marine algae. Those are also fun to put on the microscope, as well as the microscopic stuff. Yeah, not too far from the mountains, not too far from lakes and streams. So many habitats to get collections of critters from, and bird watching, and looking at large plants and mammals and everything else. Astronomy, great for astronomy in the mountains. So, for a lot of us, that's why it's worth still living here, even though it's so expensive. This thing's really pretty cool, but the slide will eventually dry up, so let's go look for something else with a little more action. Oh, look at that, dude! Yes, perfect timing. Look at that, details. So this is one of those little Stylonkia dudes. Look at that structure. Oh man, that is cool. I love oblique lighting with a little bit of dark field. So he's doing that kind of beetle thing there as well, you see? Acting like a little beetle crawling on stuff. Those little cirri act like legs. Hey Matt, how's it going? Just in time for some cool stuff. This stream tonight has had a lot of wild stuff. I've caught behavior of things I've never seen before. Found a bunch of critters I haven't seen before. I had maybe not as good a look at. This one, I haven't 100% identified it, but it's most likely this one called Stylonkia, which it's a ciliate, so it has cilia all around the body that beats to help them swim around like little tiny oars. You can see the really thick cilia fused into these clusters called cirri, C-I-R-R-I, and they use them to paddle around like the cilia with much more power. In this case, they also use them like little legs to walk on things, almost like an insect. You know, they're single-celled animals. It's just a trip to watch them. And it's got this blue-green algae kind of slithering around because they can also move. It's a colonial algae. Kind of slithering by the ciliate. And I'm using a combination of a little bit of dark field, a little bit of oblique lighting, but I can change it to more dark field, or bright field, or now here's dark field for sure. Oh. the cilia makes a nice current. The little guy got sucked in there for a second. Hydras are cool. I haven't seen a Hydra in about 20 years. I used to find them all the time in ponds and stuff, but last few times in the last couple years I looked in ponds, didn't find any of them. So I don't know if they're less common than they used to be, but I'm having zero luck finding Hydras, and they are really cool. Now if I change this exposure a little bit, you can see the particles dancing around created by the vortex of this guy. So little particles dancing around. Volvox is also awesome. I haven't seen a Volvox for a long time either, and I used to see those a lot. So I don't know what's going on, but I used to have a lot more luck finding those. And I guess conditions and everything has to be just right, because I've even looked in some similar places where I used to look and have not found any. In the last basically year of doing this show, these streams. Actually, a year and a half at this point. It's been a year and a half I've been doing this. Eventually I want to save up and get a DIC microscope like MicroAddict has, because those are killer. This picture's pretty decent with the tech I'm using, but the DIC microscopes are just awesome. Or, you know, even a what's that other one called? My brain is fried right now. It's been a long day, but you know, those start around $5,000 for a basic one, and quickly go up to easily $10,000. You start adding accessories to them and stuff. Like a fluorescence microscope would be nice too, but that's another price tag. So if I can do a quick white field, so I just can get an idea what they normally look like in most microscopes with no special settings. And he took off. Oh, now it's on the move. Come on, dude. Don't do that. Now I have to, the video game, I have to follow it. There we go. Yeah. Okay, he's on a path now. Good. I'll just stay here. Do your little path. And then you decide to change. So you can see that's the natural color of them. Kind of like a diatom color, because it's golden color there. Oh, there's that guy nodding up again. Look at that. That algae is crazy. It keeps twisting and turning, making little knot, almost like knots. And then it's sliding along its own length of the thread there. It's really cool. Look at these guys in the same field. Look at that. So that is that part of the slide. Let's keep going because this is cool, but I got way more stuff to look at. So let's Ooh, nice. There's a euglena and a nice. I've seen one of these guys before too, and I don't know what it is. It's not a blepharisma. It's a very large ciliate, and they're really interesting. But I haven't identified this one yet. But you can see it has a really long oral groove, like a gullet thing around the one side of the body with that long cilia. And as they rotate, you can actually see them ingest things and kind of spit out the bigger pieces. It kind of brings them into the gullet, and then the contractile vacuoles, the food vacuoles form at the cell membrane, and it packages into little food vacuoles for later ingestion. And I'm really happy I found this guy because I've only found it once before. Look at that. So the euglena for scale there. At the bottom right, there's a euglena. I just bumped into it. But these are fascinating. I actually have footage of just chasing this one around in kind of a rocky sample. And it keeps bringing in this, it's sucking in rocky particles and spitting them out. Like it's like, bleh. And then it's chasing around, and then there's so much debris on the slide. As the slide started to dry up, there's wave fronts of the water coming in basically trapped it into this little pocket. And before I could add some water to the slide to remedy the situation, it quickly contracted and squashed the thing into like a little box of debris, and it couldn't get out. It was struggling. I was trying to add water to it. It didn't help. And, you know, it got pinched off and it was still alive. But it looked like it had like a head and a body and a tail section all pinched off in different sections, and it was still struggling to get out. It was really, really wild. Anyway, it eventually got enough water added to the slide, and I was able to transfer it out so I didn't have to watch it get smashed. But they're beautiful, very large ciliates. This one's pretty tapered. The one I found before was really fat. It was quite a bit thicker, but had the same basic body plan and this kind of brownish color. But big ones like blepharisma are about this size, not so tapered, generally have more of a kind of tan to purplish color, and have these long lines running up and down the bodies, because these almost like striations on muscle. So it's a very distinct look, and so I don't think this is one of those, but again, I haven't figured out which one it is yet. So I'm glad I found it again. You can see that that gullet again, that oral groove on the left side there as it's poking around. You can see the really long cilia hanging out of it that are beating to bring stuff into it to help it feed. It's a really cool critter. And there's euglena again. So I'm going to give a little attention to a euglena because I like euglena a lot. Let's go zoom in on euglena. It's the photosynthetic animal. Most of the stuff you're going to see like this are plants that have chlorophyll, but this is an animal that has chlorophyll, which is really bizarre. They also have red eye spots so they can see where the light levels are, and they know where to go to make sure they get more light for the chlorophyll. So in that sense, they're what you call an autotroph. They can actually make their own food from sunlight, just like plants do. But they could still eat if they need to, and there are some euglena species I've seen that have no pigment, no chlorophyll, and they have to eat. So let me get this light figured out here. This is not, there we go. There it is. So you can see the red eye spot on the left there, and you see how flexible they are. They're quite gelatinous. where they squish themselves back and forth and into a ball, and sometimes they'll puff up and get kind of fat. There's the eye spot there. And they're just really fascinating because of this weird linkage between plants and animals on a single cell level. And you can really see the chloroplast in there. As it shifts around, they get all kind of squishy. There's the bottom, so it's kind of squished some of its body mass, even though it's one cell, it's body mass to the bottom there. And it's kind of creating the top of the body like a tendril waving around. They're very interesting little critters. It's got that beautiful red eye spot. Oh, you might be right, Teeny. I haven't seen all the Microcosmos videos yet, so I have Jim's book, which is pretty good, but it's not really encyclopedic as far as reference. It's just a really nice reading book and stuff. But I'll take a look. That could be what that little critter was, or big critter, I should say. Like so much of this stuff, you know, especially when you're out doing what I'm doing, when you're randomly getting samples from all over and never knowing from one minute to the next what you're going to find. Sometimes you go, what the hell is that? I've never even seen that before. And it's unlike certain things where it's easy to quickly classify them. I mean, it's easy to say, oh, it's a ciliate or it's a flagellate, blah, 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 blah. Oh, wait a minute. Looky, looky. There's that guy again that was swimming around in circles. That I've had squash a couple times on video. Interesting, very interesting. This is a really good sample. There's all kinds of stuff in here. There you go, he's bending over that thing. It's very thin, flexible. They can kind of crawl over stuff by squishing themselves, like that guy was doing before. Those like little slugs. Look at that. Just squished right through there. Wrapping itself around that. That's bizarre. So cool. Oh good, I got some footage of that guy too now. It's nice. But I really want some more Euglena footage because they're pretty wild. This is an embarrassment of riches. There's way too much stuff to shoot here. There's a diatom shell there. Dead diatom. Let's go back to Euglena. Where's Euglena? Where's my friend? There it is. Now it's more straightened out a little bit. Now occasionally you can see they have a very fine flagellum. Very long, fine flagellum. Occasionally I can defocus and see it, but sometimes it's really hard to find. So you just see them kind of slithering around and locomoting, but they are using the flagellum to move directionally. Perhaps if I do a little trick here, I might see a little bit better. Let's see if you can get a little bit of view of the flagellum. so far. I have captured it before, but just not really seen it now. But they are very colorful, very fun to look at. But quite squishy compared to a paramecium, which is fairly rigid. These guys are just so squishy. Whoa, that guy went flying by. Looks like somebody threw something. Okay, this is it. He's at the point now where I have to go back a level because it's so bunched up. There's way too much Z focusing going on. So what I can do is I can cheat. Do a little digital zoom here, which is not always the best quality, but it allows me to frame it better. And I don't have to worry about the focus as much. Do some more of the oblique lighting. Give the structure, this is a combination where you get some color and the dark and a little bit of 3D. Yeah, it's sacrificing too much with the zoom. I'm gonna bring the digital zoom back a bit. And here comes a rod out of nowhere. It's cruising through. Oh, a nematode right off screen. Look at that. There's a lot of activity here. Look at that guy wiggling in the debris there. Now you can see why I'm so happy with. Every time I collect at this place, I get a lot of good stuff. So there's Euglena. Oh, there's a little flagella goes whipping through there. Very nice. I'm gonna find that big dude again. I really like that big dude. Oh, another Euglena down here. That one's a little bit bigger. Quite worm-like at times. This one's a little more flat. Let's go in close up. There we go. As long as it doesn't bend too much in the Z direction, you get a nice view of it in the same plane. So unlike a flagellate or ciliate that has a lot of food vacuoles in it that are all different colors, this guy, since he's making his own food with photosynthesis, it's pretty uniform inside there. You've just seen a lot of the structures for allowing it to process its food from the chlorophyll. But you're not seeing a bunch of multicolored vacuoles inside like you get on a typical ciliate or a flagellate. So the flagellum is off screen now. It will be up here, but I'm just not seeing it. It's long and wispy. Occasionally you can see it defocused, but I'm just not really seeing it tonight. Nice linear Euglena. There you go. It's not all bunched up and squishy. favorite microorganism. Too hard to pick. This one's cool just from an evolutionary point of view. It's this weird animal plant hybrid thing, just a trip. But yeah, it's just the unusual stuff. Even some of the mundane. Paramecium is just a lot of fun. Amoeba is a lot of fun. Vorticella is really nice. Ciliate. A couple of my previous streams, you can see some tardigrades, which are always a lot of fun. What else? Gastrotrix are really cool. They look like those. They're swimming through the water, but they look like they're floating, flying through the air, like those creatures in the Matrix with all the tentacles that are attacking the ships all the time. They have that kind of motion in them. They're really cool. They're just out looking for food like little hunters. They're just eating mostly bacteria and tiny little flagellates and stuff. They're not like some kind of like a lion on the prowl or whatever. They're just out feeding on little guys. But the way they move is with a purpose and very aggressively, and they just have this cool body shape. It looks almost like a planarian or a torpedo kind of shape. But they're just really agile swimmers. They're about the same number of cells as a rotifer, about 1,000 cells. Rotifers are also really cool too. There's just so many of them. It's really hard for me to pick my favorite. And occasionally I find some new ones. I go, ooh, I think that might be one of my favorites now. But you never quite know. So there's a nice static Euglena. It's very, very still. Usually they're very active. So let's go back out and look for that big dude. I like that big dude. Where'd the big dude go? Oh, getting crystals out here. Look at all those crystals forming as the water is drying up. Salt crystals. Looks like a topographic map. So I better hurry because I'm gonna miss that guy, but he's gonna dry up. All kinds of crystals everywhere. Gotta work fast, gotta work fast. See it. It may have already expired. But that's interesting. The hell is that? What the hell is that? That might be a little cysty kind of thing or whatever. Not seeing any activity inside of it. It seems pretty dead. But it's definitely not random debris. It has a definite shape to it. Interesting. Very interesting. Gotta keep moving. Where did that dude go? Yes, you're already getting some pockets here. This guy's trapped in some debris there. Oh, I can't get out. No. Oh, no, I did get out. Okay. That was my chance to see that guy because it was so fast. Give up on that one. It's too fast. Those flagellates are crazy. So we're starting to get air bubbles as the water is receding. I can't find my big dude anymore. That was a really cool. I got some good footage of it at least, so it's nice to see it. But I don't know where it went. Don't know where it went. Oh, there's a nematode again. Hey, nematode. slowly. Ah, there it is. Got it. It's very still right now. It's just sitting there with a, looks like a little chainsaw. Whee! There's the natural color. It's kind of a brownish yellow color. Those cilia are going to town. And then I can go change the light level a little bit more. And there's the oblique lighting. Get some cool 3D structure to it. And a little more on the dark field so you get the transparency with the backlighting. It's a little more clear, white colored here, milky. But what I want to do is zoom in on this guy before the slide gets old and check out this structure. There you go, look at that. Oh man, the cilia are going crazy. It does have grooves on it, so maybe it kind of could be a blepharisma. No, this is a close look. It does have these long grooves on it. It may actually be a blepharisma. Look at those grooves. I will have to look it up. Yeah. It's just cilia are going crazy on this guy, but it's really, really large, distinct cilia. That's why I remember this one every time I see it. This is like only second or third time I've seen one. And you go down, look at the body here. And some of these cilia are basically not really moving, which means it's probably expiring. Yeah, it looks like part of it's already died. There's no motion down here, and some constrictions, which is really odd. It looks like part of it's already died. Yeah, it's, I think it's lysine. And you're seeing just the tail end of it still alive. Look at that. All this down here is not moving. But the rest of it's still alive. Pumping away. The cilia are all dead down here. And the cilia are dead here. And you can see this up here. It looks like it's slowly, this movement, it looks like it's probably slightly ruptured. Yeah, it's getting fat down here and smushy. So I think it's, as it's dying, it's rupturing and going up to the top of the body. See the constriction there, and it's all kind of smushy, oozing out here, and it's slowly moving its way up. You can see the cilia stopped beating at the bottom of that last colorful part. Also the color changed too, so that's a big change as well. So it's not long for this world. I think as it's dying, it's just not producing the same chemicals, or those chemicals are breaking down, so you get a color change. Just like, you know, your blood changes color based on chemistry. Red for oxygenated, bluish for not, purple bluish. And as part of the cell stops doing its function. It's just changing color. That's wild, man. Never seen this before. Let's see, it's holding together, but it's smushy, meaning it's starting to ooze and lose cohesion down here. Looks almost like a segmented worm down here. But all the cilia stop eating, and the color changes more pale down here now. Well, it's still twitching a little bit, so it's not totally disconnected. I just saw it twitch. Yeah, the cilia at the top is very slow now. Some of it stopped moving completely, and some of it is barely moving by the kind of oral groove area. It's slowing down. It's kind of morbid, but it's also fascinating because just like how insects can take serious damage to the body and still be alive, these guys can basically, I mean, look at this thing. It's that part of the cell is kind of okay still, even though the cilia are slowing down. But this whole area down here is just destroyed. And the cilia are getting very slow now. Very slow. So it's just about done. Yeah, see now the ooze, it's lysing slowly and the ooze is pouring out, forming that structure down there. Oh, that's rough. It's spilling its contents out. Just like a balloon squeezing out. There it goes. Whoosh. And now it's just going to slowly dissipate. But unfortunately, the slide won't last long enough for that to happen. So this is as dramatic as it's going to get. But it's still holding it together pretty well. It hasn't completely lysed and just spewed out everywhere under pressure. It's got this weird... I'll zoom out to another lower power view. You can see it's kind of still holding together, which is weird. So if you came across this, it looks quite a bit different than it did when it was alive. It'd be like confusing as hell. Like, what is this thing with these segments and the weird shape? It kind of throw you a little bit. But it is dead. Poor little guy. Glad I found it while it was still alive, though, because I wanted to get another look at it. They're pretty cool. Really large critters. I mean, you see how big the nematode is. Not too far from here. Where's the nematode here? Thought I had a nematode over there. There's the waterfront advancing, so the slide is getting dry. Yeah, that's the poor little guy. Poor little guy. You did a Native American prayer, and I thank you for sacrificing yourself for, instead of our nourishments, for our entertainment. So. Okay. So there we go. That's it for that slide. Now let's go to, that was sample B. Let's go to sample A. Coming all the way backward. That was a good one. Very good one. And I still have sample A, and then I have sample 1 and 2 from the other site. It's going to take a while longer here. Hopefully you guys can hang in there. If not, I'm going to crank these out for these samples. They'll last another day or two, but I'm going to try and crank it out tonight, because what the hell? What else I got to do? I'm not going to work on my music thing till tomorrow for the Hardware Jams contest, so it's either this or watch more Netflix, and I'd rather be doing this anyway. Let's see here. This one has quite a bit of debris. When I stirred up the silt on the base of the pool, got quite a bit of brown, you know, dark brown, black material. So hopefully there's not so much here that it obscures the view. There's quite a bit of color to the drops here. Let's see what we got. Yeah, I'm gonna probably crank through just because I want to watch a little bit of silly TV before I go to sleep. So I'll probably be another half an hour, maybe a little bit more, depending on what I find. If I find some killer stuff on this slide, I'll be on this damn slide for a while. This is a pretty epic night for these samples. Let's see what we got here. Let's see what we got here. Let all this stuff equalize out here. Got all kinds of currents for the sample. Look at that, that's wild. Look at the bubbles changing shape. Alright, let's see. I think it's settled out now. Do some scans. Oh, what did I see? Anoplius larva. Yay, Anoplius larva. This is a copepod larva, very similar to, since crabs are crustaceans as well, they also have Anoplius larva, similar to this, just different because it's not a copepod, but they have this great little jerky motion. And since they're larvae, they don't have the same appearance or a number of appendages the adult will have, but they have a red eye spot, and they're very clear, and they just have this hilarious jerky motion as they move around. It's very funny. So it's a copepod Anoplius larva, and you should have to wait for them to stop for a little bit so you can get a close-up view. I'm going to try to go a little closer and just see if I get lucky here. Sometimes they take a little break, and you can see them pretty well. Where'd you go? Where'd you go? Close, and now he's hard to find. Ah, there it is. Yay, go, go, go. There it is. You can see the gut moving around in there. And he's also, there's too much water here, so he's up and down a lot too, so I gotta keep focusing. Looks like a little monster, basically. But then they become cute little copepods. Come on, where'd you go? There you are. Sometimes you get a couple of them in here. But they have this hilarious little jerky behavior. I get seasick looking for this thing. Nice when they take a break and you get a quick look at them before they bolt. See the red eye spot at the top there? It's got basically, it looks like 6 appendages. Alright, where'd you go? Where did you? There you go. I see you. Yeah, so much movement. Unfortunately, he's not under the cover glass, so he's got a lot more leeway. Harder to find, but you got a couple of good looks at him, I hope. Let me fix the lighting a little bit. Maybe it'll help get a better look at it. There you go. Nice transparent view of the Nauplius larva. But so much focusing. Come on, dude. Tons of little flagellates in the background. Look at those particles dancing. Oh, there it goes right into frame. Oh, oh. 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 He's covered a lot of ground. It's got to keep going until you see the movement. Occasionally they do get under the cover glass, and then there's to get a shot of. There's a nice diatom there in the middle. Looks like it's already died. There's a little bit of debris in the middle of it. Let's go back up here real quick. Maybe we get a nice shot of the larva again. It might have gone to a different area. Sometimes I'll just bolt and just take off into the part of slide. Don't see it. So there's some nice little flagellates there swimming around. Alright, let's just do a little survey. Lots of dead diatoms. Lots of little tiny flagellates everywhere. Looks like that larva was the only thing good on the slide for easy viewing. Might just make another slide. No, I know there's some good stuff in here. Yeah. Let's make another one real quick. That was interesting, but not quite. Easy to find something on there. Let's back to this file here. Let's try this. Yeah, sorry, that one's a little bit crazy. Thanks for hanging in there. This is because I always do this by the seat of my pants. There's no plan, and except for certain specimens, I collect certain plant specimens and stuff. So it's kind of fun to see. This is what's out in the great big world, and you never know what you're gonna get. Sometimes you get nothing, and other times you just goes nuts. So lots of little flagellates going around. Too fast to identify. Let's do a little survey sweep again. Nice, look at those guys. Very cool. Fix the light a little bit here. Okay, that's pretty good. So yeah, lots of little dudes swimming, swimming, swimming. No larva yet. Might get lucky again. Tons and tons of, look at those little tiny dots everywhere. There's just tons and tons of little flagellates going place to place, but they're just impossible to track down. Some are really fast. Chunks of debris of something. A lot of stuff here. A whole lot of stuff. Nice one there, but he's too fast. You think the debris would slow him down a little bit, but they just go cruising right through it like it's not even there. Oh, nice. Look at that one. I've seen one of these before. Slightly slower. Flagellate, kind of like a bowling pin shape. But I can't quite tell what it is. I can probably cheat here a bit, do a little bit of a digital zoom just to get closer without having to change eyepieces. A lot of focusing. So active. But a couple decent looks at it here. Well, it's got that classic wobbly, topsy-turvy movement that flagellates have. Like that guy right there is very wobbly. And this guy's in and out of debris a lot, so it's really hard to know when it's going to come out and what level to focus at. Yeah, see a little bowling pin shape. It's very distinct. I've seen them before. Do a quick look in the white light just to see. I want to see what the true color is. Okay, so it's kind of a golden color. Get that stupid hand out of there. Again, the forest analogy. In and out of the forest. You gotta hang out and wait for them to pop out. But if you look in the little forest, you will find them. I've been to tropical rainforests in Madagascar and Costa Rica and other places looking for birds just barely peeking out of the tropical rainforest debris and bushes and stuff. Oh, there it is. Oh, there it's gone. And that's the only time you see it on your entire trip. You get a quick glance and that's it. So I'm more patient here. I can hang and get a better look at something. I don't have to fly thousands of miles away to get a quick glance at something. That's a nice little specimen, but not getting much detail there, so let's keep looking. There's another one right there, looks like. It's in the thicker part off the cover slip right there. See if I can find another one. That one's got a really funny shape. It's lumpy. Gotta stare at a couple of these things. Sometimes they, ooh, that's an interesting little cluster of diatoms there. Look at that. I believe those are diatoms. A distinct shape with a nice section to it. Yeah, those are diatoms. Look at that. They're all kind of bent and squished together. Get that fixed here. Interesting little colony of diatoms. It's not my favorite objective. One of my this stupid objective I had to replace because I dipped it in oil by accident when I was in a rush, and I just couldn't clean it. So I got what I thought was an equivalent objective, but you can see the focal range is off, so I have to go and get another replacement. It's got some distortions here as I focus through it. Interesting little guys, though. Yeah, name's Andy. You taking off, Matt? And so that's diatoms. Let's go back. It's not looking that great. Not looking that great. But interesting to take a peek at it. I'm still looking for some. There's a little Euplodes again, doing his little bug behavior. Go, go, go. Alright, I'll go look at that guy. He's fun. this guy. There's another diatom there. There's kind of a fat. You can see the rafe really well there. It's a nice comb structure. Whoa, what was that flying through there? More diatoms, more diatoms. But look at all the little things here. Fix this here, so you can see it better. Just particles everywhere, things flying all over the place. Let me zoom out on this, it's too close. Particles, particles, particles. That's a weird little dude there. What is that? It's like a flagellate that's connected. That is odd. I will have to zoom in for that, I think. It's like two little guys connected, spinning around. Like they're stuck on something. You can see over to the side here. The little, it's like a bow for bow and arrow shaped diatom shell. I was also calling it the banana earlier. And then another kind of diatoms, a little pile of diatoms right there. This guy's a trip though. Wiggle, wiggle, wiggle. Definitely spinning around. Spin, spin, spin. Not sure what's happening here. They could be colonial, they could be attached for some other reason, but they're spinning with no real sign of going anywhere. Like these things get caught on things all the time. Got caught on something, I think. It's crazy. Let's see what else is in this general vicinity. Now that I'm zoomed in. Oh, there's the diatom cruising around. Going behind the debris. Some dead diatoms. This is where that little Euplotes guy was before. Is he still here? That diatom could still be alive. There's some color in there and there's some oil droplets. So hard to say. I don't see it moving yet. But has the right color. There's that weird diatom structure again. There's an interesting diatom there. Focus through all that. It's like a diatom graveyard here. This one's cool too. It looks like a mouth, like two big lips there. I like that diatom too. You see those once in a while. What else we got? Oh, there's a little diatom swimming. Go, go, go. Oh, that's cool. See that little, just to the right of the diatom, it took off. Watch these little things. They're spinning bacteria. You see it go in and out. I'm just going to stay here. You'll see things go and spin out. They look like they're little X or Y shaped Actually, they're more like corkscrews, little corkscrews. Let's see if I can get an area where they'll stop, and you can see them stop and spin away. They're spirilla bacteria. Basically, it's the typical three main types of single or short chain bacteria. You get cocci, which are the spheres. You get bacilli, which are the rods. Like E. coli is a bacilli. And I knew that diatom going crazy there. And then you get the spiral ones, like a spirochete type structure. And they're really clever because they have flagella all along the body, and they coordinate so they spin. So it's like a spinning corkscrew going along the corkscrew axis. And it seems to be a strange structure at first, but then you watch them effortlessly just tunnel through a debris of goo or piles of other stuff. And unlike a big blocky kind of rod or a sphere, they just tunnel through right through with almost no effort. So it's a really clever shape for these bacteria to have. And they're quite quick. You see them zip around all the time. Let me just find some more here. They're really fun to watch. And there's some that are sulfur bacteria that break down sulfur bonds for energy. Some that are, there's a nice view of it there. See, it's kind of a short corkscrew. Let me find another one here. They're kind of fat. There's one right there. See, it's kind of like an S shape, but it's actually a corkscrew. And they'll stop for a while, and then all of a sudden they'll go bling, spin up and take off. Yeah, there it goes. So those are bacteria. They're actually pretty high power now. This is 400 power. So some of the bigger bacteria like that you can see quite well. And I'm not sure. I think these are just spirilla bacteria, which are basically basic decomposers. But there are some that, there's one called desulfovibrio that breaks down sulfur bonds in the mud, which is why the salt marshes smell so bad all the time. They're breaking a lot of sulfur bonds. Along with the normal decomposition, you're getting that kind of chemical smell from the breakdown of sulfur bonds for energy. And yeah, it's quite a olfactory experience in the salt marsh. Oh, there's a zippy. Go, go, go. I love the spiral bacteria. They're really cool. There's some are much longer than this one. This one's pretty short, which is why I think it's spirilla. But yeah, it's about as good a look as you're going to get at them. I could go a little higher with the oil lens, but it's not going to be a massive difference in the appearance. They're pretty small bacteria, but big enough to be seen at this power. Oh, there goes another one. Zip. They just spin and go ring. Really fun. Is that one right there? No. There you go. There are a couple of them. Zip. The problem is they often go on the z-axis as well, so they get out of focus really quickly. So it's hard to find a nice trajectory where you see it spin up and go straight off screen. But they're pretty cool. I like those guys a lot. Oh, cool. I have some friends that went to Vietnam. I haven't had a chance to go there. I haven't even been to China. I've just been to Japan, mostly from Asia point of view, which is a lot of fun. But Vietnam's a whole other thing. It'd be pretty interesting to go out there. So, not finding any big stuff to look at. Lots of cool little things. No amoeba. amoeba, no rotifers, no more big ciliates like that one I found before. Well, lots of fun little things. I'm going to try one more sample from the B file, which is really good. And then I'm going to go to the last two files, just do one slide for each to cover it. And then I have a couple quick plant things I was going to look at since I picked them and I wanted to dry out from nothing. Go to the dissecting scope for a while for a change. Maybe I'll do that after this. And then go back to the last two files. Let's take a look here. Let's see what this one has. Let's back to site B. Alright, let's focus here. So, I got a little too much water in there, it looks like. Looks like I got some water on top of the cover slip. That was dumb. That's what that little advancing thingy is there. Some little flagellates there. Finding too much on this one. That's catching my attention. Go to plants real quick. Take a break from this guy. Take a break from that and go to dissecting scope. Okay, so if you saw my thing the other day, I had some various plants from the area. I collected some stuff before, but what's fun here is I have shrub here. This one actually has, I think it has a spittle bug in it. I made a little compartment to stay moist. It's like a type of aphid. See the froth right there? It's in the junction of this branch of this leaf. And they do this so they don't dry out. It's called spittle bug. Just looks like frothy saliva. And if you move it aside, you can see who's in it. Let's see if I can find, where's my little pointer thing? I can't find my, I think this will work. This little probe. My dissecting kit. Zoom out a little bit so there's a little more breathing room here. And let's see what I got here. You can kind of see it in there already. It's kind of green. foamy matrix. Oh, he's really in there. Wish I could just move it and get it right out of there. If it had left. Maybe it made this and left, went to another place. Oh, wait a minute. No, there it is. It's over here. It's trying to get away. I see you. I'm gonna flip it over here. It's trying to get away. Hello. There's the spittlebug. This one's more like a larva, actually. Usually it's like a little aphid kind of thing, but this one looks more like a larva. See, the mouth part's going to town there. Not trying to hurt it, just trying to expose it. Come on. Boy, it's really good at evading. Flip it again. It probably doesn't like the light either. This is a pretty intense light. Even though it's a cold light, it's like a LED light. It's not too happy. Come on, I lost my spot. There we go. There you go. So it's got little mouth parts, kind of like a moth larvae or something like that. So last time I saw one of these, it was just like a little aphid thing, but this one's more like an actual larva. So this is making like a gelatinous cocoon thingy. So now we're not gonna bug it anymore. It's had a tough enough day that I already took it away from its surroundings. It's kind of fun. Let's see what else we got. So another thing out there, because it's a salt marsh, and I had some pickleweed in my thing yesterday. But what's cool is pickleweed has a parasite called dodder. D-O-D-D-E-R. Genus is Cuscuta something. I forgot the scientific name. It's Cuscuta something. And look at these little threads. So it's just little orange threads, and it has these little. If I get to the part where it's actually embedded into the pickleweed, it actually attaches little rootlets into it, basically to parasitize the pickleweed. Now it doesn't kill it. It's not a bad parasite. It just lives off of it, and because it doesn't need to make its own chlorophyll, it is a plant, but it doesn't need to make its own chlorophyll. Chlorophyll is very expensive to make biologically, so this plant figured out a way, evolved a way to live alongside the pickleweed, invade its tissue, not waste energy making chlorophyll, and let the pickleweed grow all the food that you'll eat together. And you can see how it's all kind of wrapped around here. Let me see if I can get a better angle of it. It's got its little tendrils into the tissue of the pickleweed. first time you saw it, you think, what does somebody do? They just threw some like orange like thread or plasticky crap out into the plants, but it's there on purpose. It's growing. It's a seasonal thing because it is fragile, because he doesn't have a lot of thickness or protection or waxy like the pickleweed does. So it has to grow on top of the pickleweed. I don't think it can actually touch the salt water. It would probably kill it. But it's able to live on the pickleweed and get its tendrils into the tissue and get its food. So it's with moisture. It was kind of fun. That's the first time I put the pickleweed under a scope in a long time. Here's a smaller piece that might be easier to look at. Pull off some of the tendrils here. And see, it's really wrapped on there. It's got some like little rootlets digging into it, parasitizing the pickleweed. And then just to see the stuff by itself, this is just some daughter by itself, just little threads, very fine. Pretty much like a coarse human hair. They're pretty fine, but not still soft a little bit. So anyway, that's that stuff. It's kind of fun. That's seasonal, so you only see it this time of year. And then you get the fennel growing out there. Slightly different than the one you season and cook with, but there's the fennel. Zoom out so you get a better view of it here. Very finely dissected leaves. Also, actually, very much like the daughter threads, very thin, lace-like dissected leaves, long leaves. Very, very delicate. It has a nice fennel smell. And then the flower heads, because the plant is part of the group, used to be called umbellifery, and it forms this little umbel, which is like a umbrella. Can't get this to go out any further, unfortunately. This is a small version of it here. It makes this little umbrella flower clusters that taper off in a straight line. If this were not all kind of wilted and stuff, these flower heads would spread out into basically a straight line, like I'm trying to draw with this. It's too small to go in here, but and it looks like almost like an acacia tree you see in Africa, which is a flat top. Looks pretty much flat top, and then all the branches kind of curve around to form branches that meet up at this flat top, and that's called an umbel. And that's what the fennel and plenty of other plants have that same structure. It's kind of fun. And let's see, I already showed you that plant. And this is the one I think it is. Let's see here. Yeah, this is we had these so I used to work at the children's zoo down the street from where I collected this stuff in Palo Alto. And we had this tree for the longest time. And you can see the individual white dots in the leaves when you hold it up to the light. Here you can really see it because I'm zoomed in quite a bit. But it's a unique look because you don't normally see this. I'm actually using a backlight to bring out if I take off the top light, you can just see the transmission light. And it's real distinct on this one. I think it's called a NGAIO, N-G-A-I-O tree. I'd have to look it up again, I forgot. And it has this dot, this characteristic diagnostic pattern in the leaves when you hold it up to the sunlight. It's kind of interesting. And if you flip it over, it's the same thing because it just transmits the light through it. Pretty cool. Little things like that in nature. You saw the fennel, you saw that. I have a few plants from the other day. I forgot to show on the other stream. This is... I forgot the name. Oh, damn it, I dropped a few of them. I forgot the name of the... Shit, where did that thing? I had a cone I wanted to show you. Oh, there it is. Got the cone. And variegated leaves are interesting because they're common in cultivation, but they're not really a natural formation. There's just this kind of splotchy absence of chlorophyll. And in some cases they are. This is light going through it, so it looks really weird and blown out. But if I turn the transmission light off and the top light on, you can see it's this kind of yellow splotchy pattern on the leaves. Again, I can't go too far out because the scope is magnified, but variegated leaves are very interesting. These are very common in cultivation. People just seem to like this splotchy pattern, which is not a really natural color for the most part. It's a cultivar. I thought that was kind of fun to show that because it's looks like paint spattered on the leaves. And then I had a piece of... This one started to dry up a little bit, but I did grab a piece of this fern. It started to curl up already. And on the top side you have the leathery leaves that are, you know, usually quite straight, but now it's curled up and dried up. On the back side you get all the bundles of spores. And in this case, when you get really close to the bundles of spores, this particular one has... Get close here with this probe. Hello, where are you? There you go. It has these little it's like a little umbrella, and the spores are kind of contained. Well, some have a cluster of spores like this in a bundle. Okay, this one's actually the bundle one. Some have a little bundle of spores that kind of stuck together. Other ones have like a little papery umbrella, and the spores are actually under that, so they're protected before they go. Other ones just have spores glued right on the bottom in like a flat arrangement, not in a cluster like this. So just different kinds of spore structures on the ferns, which is kind of interesting. Not as nice looking because it started to dry up. I should have shown it yesterday or the day before. They got it 2 days ago. And then there are some coast redwoods in the area. They're famous around the Bay Area, all around the bay where we had a lot of fog before the climate change with human development and settling and stuff. And so there's less fog around the bay than there used to be. But this kind of plant loves fog. It has these fairly broad, not too sturdy leaves compared to the giant redwoods in the Sierra Nevada mountains. This is some young growth, so it's a little bit softer than usual, but you can see the same pattern of the leaves. And let's see if I can zoom a little bit. No, I can't do that. Okay. Oh, I know what I can do. Hold on. I can move this up a bit so it can focus out more. There we go. And now I can... That's too far up. It's not too far, not far up enough. Let's go a little higher. Okay, that's good. And a little more light, and focus down. And it's still kind of close, but you get the idea. So it has this kind of typical spreading leaf pattern. And then you have the bark, which is mostly pest-free. Things don't really like this bark. It's very very fibrous, but soft enough to break pretty easily. But pests don't like this. You can have sawdust piles of this stuff sitting around for a hell of a long time, and they don't like to eat it. And then this is one of the cones that's opened up. And unlike a pine cone that kind of spreads out pretty wide, this one kind of just forms like little cracks. They're kind of interconnected. And then the seeds just fall right out. Looks like there are a couple seeds still stuck in there. Right there. Still one in there. Yeah. Still got some little bits in there. Didn't come out. So that's the coast redwood cones. Giant redwood leaves don't have the spreading design to capture the fog. When this firms up as an adult leaf structure, it's fanned out, so you have more surface area to collect fog, moisture from fog. Giant redwoods are formed more like a sheath. So if you look at this probe here, you would have the all along the axis of this, you would have sheathing leaves. So when the snow hits it, it just kind of falls off. It doesn't collect on the branches and break the branches off. It's just kind of a clever adaptation. That's mostly what I had for some flowers and plants left over to share. This is interesting. This is called status. It's used a lot for ornamentals. And boy, is that color not showing up right. It shouldn't be blue like that. It should be purple. The camera's out of whack. But it's these papery, you know, if you have the microphone, this kind of papery dried bracts that contain the little flowers inside of it. The flowers aren't actually the purple. These are the bracts. But it is a common ornamental item. I found it walking around the other day. Get in people's houses and flower arrangements and stuff. So yeah, that's some leftover plants. I thought they would look better on the dissecting scope, but they need a lot less power. You probably better use a camera than an actual scope for those, because they're pretty big actually. Alright, so enough of that. Let's go real quickly to sample 1 and 2 from the other side I went to. It's a salt marsh near the other marsh. One was this duck pond sailing lake area where people do windsurfing. So there might be some weird stuff in there that either came from people or from animals. Let's see here. Sorry, I've been missing some of the chat playing with those dumb plants. Let's try this one. This is the giant duck pond / sailing lake near the golf course over there in Mountain View. Swap cameras here. Alright, let's see what we got in this sample. Tiny things swimming around, but nothing too special. I didn't expect to find a lot here. I was just curious what it would be like, being that it's a pretty big water feature, lots of activity, lots of birds swimming in it, crapping in it, and stuff like that, but also human use, so stuff could fall off of people and their pets and stuff into the water, and I'm not seeing anything very interesting. So, alright. Just wanted a quick look at the sample just to see if anything popped out. There's tons of tiny little flagellates if you look carefully. out of focus there, you can see tons of little guys going around, but that's kind of typical stuff you would find here. So, sample 1 is a wash. Sample 2 might be more promising. It was more of a nature area with not a saline lake, more of a typical salt marsh, but high water flow area because it's more connected to the bay. So it again may not have a lot of standing critters in it like some of the other protected areas I collect in. I'll give it a shot anyway. Let's see. Yeah, cover slip is kind of dusty. I need to clean that off. I should have wiped it. That's okay, I can see through it to see if there's anything here. And again, lots of little dancing bits. Not a lot of anything popping out at me. A lot of those places have a lot of water flow. I would expect to find tons of critters because they're just constantly getting pushed around by the current. So kind of why I want to do these last, because they're kind of quick and dirty. Like, yep, didn't find anything there. Of debris and little tiny flagellates, pretty boring. So, yeah, that sample's a wash as well. So, that probably is a pretty good finishing point. I'm just curious how long I've been going here. Well, that can't be right. Livestream timing is way off. I think I've been going about 3 hours maybe, something like that. 3.5 hours, so that's pretty good. Pretty good run. So I think I'm going to cut it off there. Kind of a dull finish there. The plants didn't turn out as well as I want, and the water samples are kind of a wash, but luckily the stuff earlier was so good. I guess it doesn't really matter so much. So thanks to you guys for hanging in there and asking questions, and if you watch back and you miss some stuff, you got any questions, feel free to leave any comments in the chat. I mean in the comment section and things like that, and I will do my best to answer them at some point. Stay tuned. I will be doing, I got 2 streams back to back here, so I probably won't do one for maybe later, till later next week. I gotta do some music stuff and some other things for work and all that, so it was fun to do it 2 days in a row, but I gotta take a break. So but once I'm back on the schedule now, I'll probably be doing a couple a week if I can get the samples in time and keep getting enough material, because without the material, it's you know, just kind of grabbing. It's been kind of dry lately, so I used to be able to get water samples anywhere from planters, from gutters in the street, puddles and driveways, all kinds of stuff. But with no rain, I have to go hunt out all the water sources I can find. So it just takes a little more work to get it. And I'm getting good samples, but it's not as convenient as it was when I could just go outside and grab something real quick. So anyway, that's do it. And again, I appreciate it. Everyone showing up and interacting, questions, and all that good stuff. And I will see you soon. Thanks again.